Okay, so we're going to start off with RNA processing and translation. First, let's review. The bases of RNA are the same as those of DNA, with the exception that RNA contains. So if you look at the choices, you should be letter B is the correct answer. Uracil, so RNA contains U's instead of thymine, T's, right? So the letter T does not exist in a molecule of RNA. So if we were going to make a molecule of RNA, right, we already looked at transcription is reading our template strand of DNA and then creating a molecule of RNA using RNA nucleotides. So if this was our little sequence of DNA, what is the RNA strand created after transcription? So if we're reading this, what RNA polymerase is going to do is look at the A, right? And it's going to say, it's not going to say anything, but it's going to add the nucleotide that's complementary to A. So that would be, I know you guys want to say T, A's and T's, but we're working with RNA now, right? So we're not using T's, we're substituting a U. So the very first base that we're going to add to when we make an RNA molecule is going to be U. So let me just show you the answers here, right? So A, the RNA molecule in red will be U. The DNA, if the DNA template strand has a G, then the complementary is C. If there's a C next, then the complementary is G, G, C, T, A, T, A, A, U, right? Because we don't have T's in RNA. G, C, A, U. So this is going to be our RNA strand, right? So it contains the complementary information from our template. We read the DNA template strand. We put complementary nucleotides in. And remembering, since this is RNA, we're leaving out the T's. All right, so this is an RNA strand. It's complementary to the DNA. Notice how uracil, right, is substituted for T's. All right. So having understood this, let's look at how RNA is processed to messenger RNA. So the little m stands for messenger. And let's start with our, so these 1, 2, 3s, this is also listed in your outlines. We're going to go in the order of your outlines. So number 1 is a poly A tail. So let's just recap real quick, right? So this is our DNA molecule. There's introns and exons. I'll talk about that in just a second. But this RNA in red is our RNA transcript, right? This is after transcription happens. So you should understand that the nucleus is like a safe place for molecules. So DNA is sheltered inside of the nucleus. The cytoplasm is a very dangerous place for molecules. It can get destroyed easily. There's a lot of chemical reactions happening. So DNA never leaves the nucleus, right? It doesn't want to get harmed. So what it does in this process, right, we've evolved this process to create a complement of one of the strands. So we're taking that same information from DNA and we're putting it into a molecule of RNA. RNA is dispensable. You are making tons of RNA every day. You're destroying tons of RNA every day. So RNA is just like sort of a disposable molecule. But while it's working, right, it's taking the information from DNA and it's going to travel into the cytoplasm to make the protein. And so the DNA can stay inside and this RNA molecule acts as a messenger, right? It's going to take the message from DNA. It's going to take that message to a ribosome in the cytoplasm where proteins can be made. So in order to protect the messenger RNA for that short period of time that it's in the cytoplasm before it ultimately gets destroyed, is 2 things on the end in yellow here. So in your outline, the first thing is our 3' poly A tail. So here's the tail. The RNA molecule has the same 3' and 5' ends as DNA. Okay, so on the 3' end we have a long A, just lots of A's added to it. So that just kind of buffers this end from getting destroyed from this side. The other side is called a guanine cap, 5' guanine cap. So there's a guanine that's put here. It's a special guanine. It also prevents the RNA molecule from getting destroyed. And the last thing for processing is a little bit more interesting. So it turns out that your DNA and in return, because the DNA has an RNA, there are pieces of the coding sequence that are not used in making protein. So you see there are two kinds of regions. They are called exons and introns. So exons are the regions that get expressed, and introns are the regions that go in the trash. That's how I remembered it when I was a student. Exons get expressed, introns go in the trash. So in this processing, the splicing step, number 3, you're taking out the introns, they go in the trash, and you're putting those exons together. So all the exons are spliced together. This is a film editing term, and you get your complete messenger RNA at this point. So this messenger RNA where all the introns are removed, the 5' cap and the 3' poly A tail are on. Now it is a fully mature messenger RNA molecule ready to leave the nucleus to go to the cytoplasm and incorporate with a ribosome. All right, what happens? How do we read RNA? So this is the interesting part. In order to get proteins, sorry about that. In order to make proteins, proteins are a completely different language, right? Proteins are amino acids. How do we take the code from DNA and RNA and make amino acids, right? Or sorry, make proteins or string our amino acids together? Well, there is a group, so the biology, right, life evolved a method of using codons. So every 3 nucleotides in a row is 1 codon. And a codon is a code, right, that codes for 1 amino acid. So you can see in this RNA molecule, there are 7 codons. There's 21 nucleotides in a row, but when you group them in 3s, we have 7 codons. And so that means that if 1 codon codes for 1 amino acid, then 7 amino acids are coded for here. Okay, and the sequence, right, codon 1 is next to codon 2. That means amino acid that this is coding for, the codon 1, is going to be next to the amino acid coded for in codon 2. And so now you can see how the letters of RNA can dictate what amino acids go next to each other. So let's look at translation. Number 1, genetic code. This is the genetic code. The genetic code is a universal code, so which is pretty amazing. So you can see that scientists have worked out what every single possible codon there could be, which is 64, and exactly what each variation of 3s could code for. So the words here, proline, serine, leucine, these are amino acids. Those are the amino acid names. You may have heard of tryptophan. Tryptophan is also oftentimes comes up around Thanksgiving. It's supposed to make you sleepy. It's the precursor for serotonin. But anyways, just so you know that these are all the amino acids. We have 20 amino acids that we need to make all the proteins, and far more codons. So if we have 64 codons and 20 amino acids, that means that some of our amino acids have more than 1 codon. So proline, threonine, serine, you can see there's 4 codons that code for serine. That's what we call a redundancy in the genetic code. So the universality of this is that everything on Earth, every living thing, whether you're a plant, an animal, a worm, a bacteria, a yeast, slime mold, an amoeba, all use this code. So this is the basis for putting genes from one organism into a completely different organism, and that gene being able to be expressed is because DNA is the same language for all of life, and the codons that you see here, this genetic code, is the same for all of life. So 1 codon codes for 1 amino acid, up to 3 amino acids, all 20 amino acids, TNA or mRNA. The answer is 1 amino acid. So 1 codon only codes for 1 amino acid. Let's see if you guys can understand that. So if we just created a codon, CCU, right? CCU. CCU only codes for proline. One codon codes for one amino acid. So CCU doesn't code for anything else. It just codes for proline. Okay. There is a redundancy. There's more than one codon for proline, but there's only one amino acid per codon. Okay. All right, so let's look at this example I was talking about, right? So this is a tobacco plant, which is expressing a firefly gene to glow. This is because the glowing mechanism, the protein that actually glows in fireflies, the gene for that, the tobacco plant, right? A plant uses the same code so it can take that information on the DNA, make the protein successfully, and express the protein. So there you go. This is human insulin. This is made usually from yeast. And then this is mice that are glowing green. This is a gene from a jellyfish. It's called green fluorescent protein. So the applicability is that we're using the fact that all of life uses the same language, and we can start to sort of cut and paste different genes to different organisms, and they can, express these different genes, and you get very interesting results. All right, so let's look at translation. So you have this step here, right? So we went from DNA, we transcribed it to RNA. RNA is going to get translated to amino acids. We saw that the group of 3 nucleotides on RNA is going to code for one amino acid. And the sequence of our codons will dictate the sequence of our amino acids. So there is a very important molecule that's in the middle step of all this. This is transfer RNA. So number 3, this molecule is the link between RNA and the amino acid. So this is how it works. There's a special anticodon region here. This anticodon is 3 letters that is complementary to the codon. Then there is a place for an amino acid to bind. It's the amino acid attachment site. So let's look at how this works. Here is part of the ribosome. Here is our messenger RNA laying inside or on top of one of the ribosome subunits, and this is our first transfer RNA. So notice here, right, there is a matching area. The red codons of RNA are binding with the anticodons of our transfer RNA, right? Because A is complementary to U is complementary to A, and G is complementary to C. So these hydrogen bonds, they're pretty strong in the fact that they can coax this transfer RNA to be stable in this position. And the other portion of transfer RNA that's really important is the amino acid binding site. So the amino acid that the codon codes for is going to be hanging out right here. So this molecule transfer RNA recognizes the codon, and it brings the correct amino acid to the ribosome. So step 2 here is that the complete ribosome is assembled. Okay. So what happens is, think of the ribosome as a 2-seater couch, right? I can see one seat is taken up, another seat is empty. I'm not going to have you guys memorize the P site and A site. But what happens is another transfer RNA will come in. Let me flip the. All right, so imagine that a transfer, let's actually go back and just use your imagination. So this empty seat, another transfer RNA will come over. It will recognize the codon with its anticodon. It will bring another amino acid to this ribosome. The ribosome will then create a bond between this amino acid first in that first seat to the amino acid in the second seat. The empty amino acid, so this gets cut off, it gets put onto the second one. This is now empty, it's going to get kicked out. Okay, so take a look what's happening here. We have a little chain going. We have an amino acid, this diamond is coming in, the anticodon. So what's causing this transfer RNA to come in? It's the recognition. The anticodon is going to be drawn to the codon. When there's a binding, okay, look what's happening here. The circle is going to leave this transfer RNA. It's going to bind to the diamond. So this is a chemical reaction, right? We're taking the amino acid off the transfer RNA, we're putting it onto the other amino acid, so we're creating the peptide bond. This keeps going, okay? So we can start here. We can see there's one seat. We have a little chain going. This is what we're looking at the other side at the previous slide. This diamond comes in. Oops, sorry about that. Diamond comes in. We get a little bit of a chemical reaction where the amino acid comes off and attaches to the new amino acid. And then what happens is this empty transfer RNA gets kicked out. And this will move over and the whole process starts again. So you can think of this as the ribosome is just traveling along the messenger RNA molecule. Two transfer RNAs come in, recognizing the codon with their anticodon region, and the amino acids are hooking up to each other as they come in. Okay, so there's two codons that you want to know, number 2 in your outline, which is our special codons. So there is a start codon, which starts the whole process, and then a stop codon that stops the whole process. So you can see the word stop codon here. So the ribosome knows, and the transfer RNA knows to stop building the protein when it hits that stop codon. The start codon, let me actually go to that image. There's a start codon. So the start codon is the codon that signals to start translation. We also say initiates translation. So start codon is always in the beginning, and the stop codons are always in the back, okay, at the end. And number 3 in your outline, I just described for you the transfer RNA molecule, okay? The steps in translation in your outline, initiation is this step where we're building our ribosome with our first transfer RNA at our first codon, our start codon. Elongation is the step where we are slowly, well actually this is pretty quick, but we're building that polypeptide chain within the ribosome. And then termination is reached when you hit the stop codon. So those are the 3 steps. All right, so this is a little bit of a review of everything. First step, transcription. So transcription means we're going to take RNA polymerase to build RNA. This large yellow enzyme, which is RNA polymerase, will unwind a little piece of our DNA, read the template strand, and then build a molecule of RNA. Remember those complementary letters. If there is an A on DNA, there is a U on RNA. If there's a C on DNA, there's a G for RNA. Remember that once we have our RNA, we have to process it. So the introns go in the trash. We're putting on a poly A tail and a 5' guanine cap so that the RNA will not get digested immediately when it leaves the nucleus. Once the RNA comes into the cytoplasm, it's going to start to assemble with a ribosome. Okay, so first we have to initiate our process of translation, right? We're going to have a start codon on the messenger RNA. We're going to have an anticodon on our transfer RNA and also have that first amino acid on our transfer RNA. Then we're going to elongate. So that empty seat is going to be taken up by another transfer RNA. At the ribosome, we're going to build our amino acid chain. And the amino acids are placed one next to each other because the codons tell you which amino acids go there, which amino acid goes next, which amino acid goes next. And so the sequence of amino acids is dictated by the sequence of codons of RNA. Okay, and when we hit the stop codon, the process of translation stops, the polypeptide is released, and we have a new protein. Okay, so here's our concept check. An organism's genetic information is stored within the sequence of blank. This information is transcribed to a sequence of blank, which are then translated into a sequence of blank. So your information is stored in DNA, transcribed to RNA, and then translated to amino acids. So the last choice is the correct answer. All right, let's see if you guys can rank these. If you want to pause the video to think about it, I don't want this video to get too long, so I'm just going to tell you the answer smallest to largest. So if you look at your choices, a nucleotide is going to be the smallest because it is only one unit, one building block of our genetic material. The next would be codon. Codon is 3 nucleotides, then a gene, then the chromosome. There can be thousands of genes on a chromosome, right? So chromosomes are very, very long. All right, we'll stop there and talk about mutations in the next video.